At Nautilus Biotechnology, we're developing a single-molecule proteomics platform capable of quantifying the entire proteome. The scale of the proteome is incredibly vast. In humans alone, there are roughly 20,000 genetically encoded proteins and millions of proteoforms. So how do we plan on enabling researchers to efficiently quantify the proteome? We've designed a novel solution that matches the scale and dynamic range of the proteome. We start with a simple single-molecule library preparation technique that prepares billions of individual protein molecules for automated deposition at distinct coordinates on 10 billion landing pad arrays. Then, we iteratively probe the isolated proteins with proprietary multi-affinity probes, and use a machine learning-enabled framework called Protein Identification by Short Epitope Mapping, or PRISM, to identify each protein molecule. The strategy behind PRISM is similar to one you might use to win the game Guess Who I Am. In this game, you try to identify one character amongst a broad set, using the fewest questions possible. You can imagine that you could play this game by asking very specific questions. Are you a lizard? Are you a dove? Are you an ape? And so on. However, unless you get very lucky, this strategy would be inefficient and would force you to ask many highly specific questions before getting to the answer. Instead, you could use a strategy like the one underlying PRISM and ask general questions that eliminate many possibilities quickly. Are you a farm animal? Do you have wings? Are you a mammal? With this technique, you could quickly and efficiently identify a character by gathering more information over a shorter series of questions. The way this manifests in PRISM is we probe each individual protein molecule on our arrays with multi-affinity probes, which are designed to target short peptide sequences within proteins, instead of individual specific proteins. Based upon its amino acid sequence, each protein has a predicted probability of being bound by each of the hundreds of multi-affinity probes. Each time we probe an immobilized protein, our machine learning algorithm gains information that it uses to boost the confidence in that protein's identity. There is information gained both when a probe binds and when a probe doesn't bind. Zooming in on two identities that seem probable by the end of these initial cycles, we see how the probability of a given identity changes with continued cycles, depending on predicted probe binding in each cycle. By the end of many cycles, it is far more probable that we have protein A than any other protein, and our platform thus identifies the protein at this landing pad as protein A. This same process happens simultaneously at each of the 10 billion landing pads on our arrays. We believe this approach will result in protein identification across substantively the entire proteome. Thus, the algorithm efficiently identifies the billions of proteins on the array based on the compatibility between expected and observed binding events over many binding cycles, using different multi-affinity probes. This is like asking broad questions in Guess Who I Am, because the multi-affinity probes provide information on many proteins at once during each binding cycle. Importantly, multi-affinity probe binding stochasticity makes it so there is no canonical fingerprint for each protein. Instead, we ask the algorithm, given an observed pattern, what is the probability of protein X? This makes the platform robust to stochasticity. Simulations show that between 200 and 300 multi-affinity probes are sufficient to identify substantively all of the proteome. Once we've decoded the identity of each protein found at each landing pad, we can determine abundance by counting how many times a protein is identified. We thereby quantify proteins across the vast scale of the proteome through the identification process itself. With our hyper-dense arrays, multi-affinity probes, the PRISM methodology, and advanced machine learning, we anticipate enabling unprecedented access to the proteome for scientists in a wide variety of research fields. This will empower advances in basic biology, biomarker discovery, therapeutic development, and much more.